Hi, I'm Brian Herb from the University of Maryland School of Medicine in Baltimore. I'm very excited to be part of this special session, and I'll be telling you today about NeMO Archive, which is our part of the BICCN to store and make accessible BICCN data, and also NeMO Analytics, which allows users to explore BICCN data alongside other published datasets and their own data. As you have seen in this session, the BICCN has done amazing work to try and understand the composition and function of the brain at a cellular resolution. This work has created an impressive breadth of transcriptomic, epigenetic, and anatomical data across the brain, and there's clearly been an enormous effort to integrate and analyze the data in new ways. I've highlighted 3 papers from the speakers today to illustrate both the consortium effort needed to undertake these efforts, but also, as we see from these plots of single-cell data, that these papers advance our understanding of cellular composition and identity within the brain. This vast amount of data and level of analysis may be daunting to the reader, and my talk today will explain how NeMO Archive helps users access data in a meaningful and organized way, and also NeMO Analytics allows users to explore the data and compare their own data to published results. These BICCN projects are very impressive, with the depth of data and also the complex analysis that's associated with them, but also presents some challenges for an investigator who wishes to follow up. And so, to mitigate some of these challenges, we want to make sure that we provide easy access to raw data and associated metadata. We want to facilitate the easy transport of massive amounts of data to compute environments. We want to make sure that all analysis files associated with papers are openly accessible. And we also want to be able to explore the data within the space of published analyses. So for example, we saw in the last slide a couple single-cell data sets where that analysis of that clustering actually represents relationships between the single-cell types. And it'll be very important to build on these conclusions and be able to explore the data within this single-cell space. So the NeMO Archive is set up to address some of these challenges. And as this is an evolving archive, we already have a data volume of about 150 terabytes and a little over 600,000 files. And what you'll find on this is a wide variety of both data types and also data from various brain regions. So as we walk down the left-hand panel, we see that data is stratified by both grants, so the lab that is generating the data. We can stratify by organisms, currently mouse, human, and marmoset. Anatomical structures is a key facet, so we can actually go through and select different brain regions that we really want to go into depth with and study. Library methods and technique are more essentially what type of data we're dealing with, and modalities is a little bit of refinement of that. So it could be transcriptomic data, epigenetic data, and it'll be growing as we gather more data into the system. So for example, if I wanted to do a faceted search and my interest is in open chromatin regions in the mouse motor cortex, what I can do is go through the left-hand side. I can select out mouse. I can go through the anatomical structures and look for the motor cortex. And then I can finally refine this by looking at ATAC-seq data only. And when I do this multifaceted search, what I pull up is a number of samples that meet my criteria. And once I do go through this searching, then I can just simply add these to my cart, and the data is available for download. Now, to most users, this might be a bit of an overkill in terms of how much data we're dealing with. So already 77 gigabytes might be too much for the normal laptop computer. So a massive added benefit to having the NeMO Archive is that all of the data files, while it's also on a server at University of Maryland, it's at the same time on a Google Cloud instance. So what this means is that all the data is actually already stored in the cloud environment, and it can be exported to Terra, which is a computing environment over at the Broad, and which also uses Google computational resources. So almost instantaneously, this data can just be moved over to the cloud computing environment at Terra. So to give you a quick walkthrough of what they do, and again, this is set up at the Broad Institute, a normal user journey would be to upload the data. But again, this is all uploaded directly and already there for you. So one nice added benefit is that all the BSCCN data, free of cost to the user, is held within the Google Cloud. And then, based on their years of experience with pipelines, there's a whole host of various analysis tools that you can move the data through. So from basic QC all the way up to single cell pipelines, you can move the data through this, and then at the end, download your results. So just as a quick review, on the left-hand side, we see the architecture of NeMO, and we see that in the bottom left that the files do sit on our archive at University of Maryland, but they're also simultaneously in the Google Cloud platform, which again can easily be ported over to Terra for analysis. Over on the right-hand side, to give you an idea of a couple of single-cell data sets that are already on the NeMO Archive, we have millions of cells that are from various grants and various labs across the BICCN consortium. These also represent a multitude of brain regions, as you see at the bottom. So already a researcher can go in and make meaningful comparisons of various brain regions on a single cell level. Now, just to highlight how this data archiving looks, we take an example from the mini atlas paper, and we have organized on NeMO Archive both the raw FASTQ files, but also we allow the authors to have a full range of any and all analysis files they wish to associate with the paper. And to simplify access to these files, the raw analysis and files were organized into BD bags. So think at this point like a fancy zip file, but we'll explain a little bit more of that later. And we stratified it by the 9 data sets that were generated to form the paper. So a quick dive into what a BD bag is. First off, it's just a general tool for assembling, sharing, and tracking data. And the tracking data is an important one because NeMO also assigns unique identifiers to different data sets. And that's very important for tracking the data throughout publications, and it gives you something to cite in the future. And at its most simple level, a BD bag is a collection of files, and that could be like a zip file. However, because in the case of some of these papers, you get in upwards of terabytes worth of data, what these are actual links to the data on the server. So when you download a BD bag from us, it's essentially just a list of all the files that are associated with the paper that are on our server, and you can easily retrieve them through BD bag software. And again, this can be augmented with metadata to facilitate data tracking, which is very important as we move forward and keeping track of the data that we've generated. And very quickly, the BD bag itself, there's a separate software for it, and when you open up the downloaded file, it simply opens up like a web browser. You can select the files that you desire. There's also a Python command line tool for it as well, so you can programmatically derive files. So what's in the mini atlas BD bag? So again, it's been associated into 2 different things: raw data analysis files. And as you see on the table on the right, there's 9 distinct data sets that have been generated. So that's how it's organized. And one thing that's nice, if you allow NeMO to organize your data and create BD bag for you, there's a single web link that you can list in your paper. It's much like how people used to do for geo entries or SRA entries. And readers then can click on that link and then have access to all your files directly. And so when they do click on that link, there's a web interface that opens up. And this is the main landing page. So you see at the top we have the unique identifier. And then again, that can be used for citation purposes. And this will be the overall BD bag. So if you did click on this, this will give you a full listing of all the raw and all the analysis files. And this can be opened up with that BD bag software. Now, to allow the user a little bit more flexibility, we also break this down into different tabs. So you can look at just the raw data or just the analysis files if you desire. And if you clicked on one of these tabs, you now get into all those different data sets that we were talking about. So there would be 9 different entries in this case. And if you clicked on just one, say for instance, the ATAC data here from the Salk Institute, you will again have a unique identifier for that data set. So you can, in fact, dissect out different data sets that you wanted to publish on in the future. So You don't have to just simply cite the mini atlas paper. You can actually be much more specific and give credit to the exact data set that you're using. And then a nice example for the analysis files, there's actually multiple ways to access the file as well. So the source data URL is actually a link back to our browser. And when you click on this, you can actually look at the listing of all the analysis files and click out and simply download directly the ones that you want. And this is very helpful because some of these data sets have thousands of individual little files. And you can actually go through this instead of downloading the whole bundle. You can just go through and selectively get the analysis file that you want. So if we look back on our challenges, hopefully NeMO Archive does address many of these things. So we've tried to make the data easily accessible either via the data portal or have a BD bag associated with each paper. And again, with the compute environments, the beautiful thing is that a lot of the heavy lifting of storage is done. It's already in the Google Cloud. The Terra platform is absolutely wonderful for doing data analysis, and essentially the data is already there. And you'll be surprised that the Terra platform is very economical. They just simply pass on the cost of the Google instance. And access analysis files associated with papers. So we're really trying to make an effort that if a reader goes in and is very excited about the data in a paper, they can go instantly into the BD bag and grab that data and also the analysis so they understand that paper in context. So the last little arm here, how do you explore data within the space of published analysis? So in the past with various genomic papers, you may get a list of DEGs or some, very static tables. But what we really want people to do is be able to dive into the figures and understand, say, like single cell data within the context of the figures as they're presented in the paper. So now we'll talk a little bit about how NeMO Archive dovetails in with NeMO Analytics. So again, all the wonderful data from the raw data to the analysis files are going to be stored on NeMO Archive. And with NeMO Analytics, what we're going to be doing is giving the user tools to be able to reanalyze the data and also be able to compare different data sets. And with NeMO Archive, we have the overarching goal of trying to support as many of the BICCM papers as possible. And I'll walk you through two examples, both the cross-species paper and the mini-atlas paper, where we've done a lot of work of trying to make the paper come alive in the NeMO Analytics website. So on the left-hand side, we actually have the T-scenes from the paper. And in our portal, we've recreated those T-scenes, and you're able to go through and individually look at single genes and their expression in the space of these various clusters. And so that kind of recapitulates what's in the paper, but we can also take that data and analyze it in new side-by-side ways. So I'll walk you through in the next couple of slides various things of looking at single genes and comparing different cell clusters. We have now a new epigenetic browser where you can take a look at in this case, ATAC data and comparing. And you can also do a lot of differential gene expression analysis. And one very exciting thing in the end, being able to project various data sets into another. So for the epigenetic data, we're working with the EpiViz team down at University of Maryland College Park to both display epigenetic data in a browser form and also have dynamic plots and displays. Now, to provide an example of how the epigenetic data can be mixed with the transcriptomic data, here's an example from the cross-species paper where they have taken a look at individual either excitatory neuron subtypes or inhibitory neuron subtypes. And as we work down the panel, you see first the gene expression profiles of every single individual excitatory group, and just below that you have the tracks from the ATAC data. And so you can quickly see where things are higher expression, low expression, and its relationship to open chromatin. And the same treatment is done for the inhibitory neurons. Another example, this is now from the mini-atlas data, kind of a similar idea, and now we're dealing with mouse data. So here we have GAD2 expression, and you see that in the overall cell clustering. And we can compare that directly down at the bottom with ATAC open chromatin regions, or in this case, DNA methylation levels. And you can see how that relates to the gene expression up above. There's many other tools that we built into NeMO Analytics. So we've already taken a look at the EpiViz tool. In the bottom left, we have a very exciting single-cell workbench. So you can actually go in and start a new analysis all the way back from scratch. So any data set within NeMO Analytics is available for this. Or you can actually pick up an analysis directly from where it left off in the paper. So in this example, we have a clustering from the cross-species paper. So this is just the human data. And we see GAD2 expression in the context of the cell types. But you can also take two different clusters and compare them. So in this case, we see in the upper right, astrocytes versus endocytes. We can also take a look at major markers that distinguish the various clusters and display them as a dot plot. So this last aspect of NeMO Analytics I would like to show you is a very exciting part where you can take patterns that are existing in one data set and project them onto another data set. So in this case, we're taking a look at human cortical development. And in the top panel, we have a data set from the Kriegstein lab. And we see within it, we have the excitatory neurons lit up by SST07-VII. And within a separate data set in the macaque from the Allen, we actually show that SST07-VII is enriched in the cortex and cortical plate. So what we're going to do here is perform a projector integration between two data sets. We're going to use the Kriegstein dataset as a reference. And once we run cogaps on it, we're going to identify different patterns within this dataset. And in this case, pattern 6 identified the radioglia. Now we can take this pattern and we can project it into a new dataset. For here, we're going to be using the macaque. And we see along the y-axis of the figure on the right that pattern 6 is actually enriching for cells of the ventricular zone. And it makes good sense because that's where the majority of the radioglia cells lie in the macaque brain development. As a proof of principle, we're going to also take a look at another pattern within the Kriegstein dataset, in this case the excitatory neurons. And when we project these into the macaque data, they of course light up the cortex region, which is where the majority of the excitatory neurons reside. And so we can take a step back and think of this as a wonderful discovery tool and take advantage of all the patterns that we find within the BICCM projects, either throughout development, across cortical or across brain region, and we'll be able to take all that hard work and analysis and project it into new data sets for discovery purposes. So I'd also like to take a moment to acknowledge all the hard work that's brought into this across NeMO Analytics and NeMO Archive and all of our collaborators. Thank you very much.